What's the microscope? Then the microscope obviously is a device that is going to help us to see very small images. There are two main things that you need to know about microscopes: parts of the microscope, and another thing is going to be properties of the microscope. Parts of the microscope are the ones that we can see right here. Right, we're going to have the ocular lenses. And there are other lenses, this one and this one and this one right there. But these ones are going to be called objective lenses. Whatever that you want to be looking at, you're going to put it right here. And the lenses are going to magnify it for you. So then how many times this object is going to be magnified before it actually gets into your eyes? I think it's going to be magnified twice. Right? One time by these lenses and one time by these other lenses. The ocular lens is fixed, meaning this always magnify this 10 times. And this one right here is not fixed. This part rotates. That's why that is called the rotating nose piece. Why you can rotate this? Because that's going to allow you to change the lenses, right? So then you can change this one to this one or this one to this one, right? By moving the rotating nose piece, right? Which is this part. Let's say you are using this lens right here. This lens is 4. So then how many times you're going to magnify before you actually see that with your eyes? Then it's going to be magnified 40 times. The 4 times right here with the ocular lens times the 10 times right here by the ocular lens. So the objective lens is going to be 4. The ocular lens is going to be 10. So therefore the total magnification is going to be 40. If you use this other lens right here, so let's say, for example, this one is going to be, let's say 40. So then if this lens is 40, then what would be the total magnification? 40 times 10, that would be 400. Okay, and let's say you change this one to this one. Let's say, for example, this one right here. Let's say that one is 10. So then instead of 40, that would be 10. So then what would be the total magnification? Then the total magnification would be 100. So then the total magnification that you're going to get in your eye varies depending on the lens that you're using in the objective lenses. And the total magnification that you get again is because you multiply both lenses, and that gives you the total magnification, as you can see right here. Okay, so then going back to this, we have in here the ocular lens. This is the rotating nose piece. These are going to be the objective lenses right here. You're going to put a slide in here like this, which is a piece of glass. Now, this is not in the air. This is going to, obviously, it's going to sit in this plate. That is going to be called the stage, which is what you have right here. In addition to that, what other parts you have in this microscope? Well, you're going to have, for example, this. This is the arm. This is what you use in order to hold the microscope. In addition to that, here you have set of knobs. This one's right here. So this is one set of knobs right there. And the other knob that you're going to have is this one. Right here, you have 2 knobs, the top and the bottom. These 2 knobs are going to move the stage back and forth. So to the front and to the back like that. These other 2 knobs in green, you have one big one, you have a little one. The big one is going to be called the coarse adjustment knob, and the little one is going to be called the fine adjustment knob. The coarse adjustment knob is going to move the stage like this, fast and big movements. So you can move the stage up and down very fast. The fine adjustment knob is going to move it like this, slowly, like this, like this. This is the light source. This is the base. Down here, you have the condenser with the diaphragm in there. These lenses are going to have different values, right? 10, 4, 40, 100, etc. I know it's not that clear here, but they have a line. This one should have a white line. This one on the back should have a red line. This one right here should have a blue line. And this one right here should have a yellow line right there. Yeah, so if it's red, right, that usually magnifies 4 times. The yellow is going to magnify it 10 times. The blue is going to magnify it 40. And the white is going to magnify 100 times. So this one is going to be the objective lenses. Now remember, the other one is the ocular lens. This one is always 10, right? This doesn't change. What are the properties of the microscope? Properties of the microscope are going to be magnification and resolution. Or let's put in here detail. Okay, so that can help you identify that. So you're looking at a microscope and this is what you see. The image is blurry. Can you see something in there? No. What good is a microscope? That it can magnify things, but you can't see it because it's all blurry. Property of microscope, one of them is to make it larger, but the other property is resolution, which is the ability to see the detail. Right? You're supposed to see the detail. Right here you have table, low, medium, high. Right, low is 4, medium is 10, high is 40, and oil immersion is 100. So we scroll down right here. Let me see. We have focusing of the microscopes, and you're going to get some slides. And there you're going to have a letter E. This is basically for students to begin to practice with a microscope. After students practice with the letter E, and then they get a slide. This is one cell right here, right there. This is another cell. You have another one right here. But I want you to see one thing. Your heart, your liver, those are three-dimensional structures, right? So you can imagine that you have in there cells and structures at different levels. And that's why you also have, something to practice, which is going to be called the depth of field, right? So because remember, you have to understand that if you look at this from top to bottom, some cells are going to be here, other cells are going to be down here, other cells are going to be down here. So you need to understand that, yes, you may see it sort of flat, but it is not, right? Because remember, these are three-dimensional structures. If you look over here, then you can see that this is down here, right? And this one is darker, right? Why is darker? Because that one is in superposition of the other one, is on top of the other one.